First of all, I would like to thank Tom and Steve for organizing this committee, this meeting, and also for inviting me. It is truly a great honor and privilege to speak on this occasion to celebrate Gobin and his scientific career. And for me, it is especially a privilege and honor because I worked with Gobin. I witnessed a great man, great mentor, and great scientist at work, and I've benefited enormously from my interaction with him. And in particular, I started my own lab from the summer of the year 2000. I joined Gobin's lab from late 1997 and left in the summer of 2000. But because of the physical distance, short distance between Albany and Cambridge, so I commuted back and forth so frequently, including a significant amount of time I spent with Gobin in the fall of year 2002, and many times he and I would have lunch and dinner together, probably for like 2 months of time in that stretch of time. Many times at Gobin's expense. One of the things I think many people know is that every time I raise the question, Gobin, can I pay the bill or can I pay the tip? Now he would get very upset. So I tell you a true story. At only one time, I was fast enough to grab the bill before Gobin realized it, and he was in a hurry of taking out his wallet and handed over a plastic card. But instead of being a credit card, he handed over his MIT faculty card. Obviously, the waitress is not going to accept a MIT faculty card as a payment. So I was glad, and Gobin, thanks very much for everything, and again, congratulations on this honor. So today I thought I would like to tell you one project we accidentally stumbled upon, but we have a whole lot of fun working on it. So I'd like to just probably give you this, again, just one project ongoing in my laboratory. This has a lot to do, again, with the polymorphic and the dynamic properties of RNA that Dr. Wells mentioned about DNA, so I'm focusing on the RNA. Now, the polymorphic and the dynamic properties of RNA is probably best for at least one feature of this is the fact that if we have a one RNA sequence, and this sequence might be able to fold into different conformations in the subpopulations, because all of which can be characterized as one phenotype. And as such, perhaps we can isolate a particular subpopulation or conformation and going through this so-called thermodynamic pathways of folding, refolding, in order to either gain other population-wise or maybe we're lost if we happen to pick up something that is not as stable as the other types. But all of these conformations are connected through this known so-called folding and unfolding pathways. As a matter of fact, we don't have to even go through this folding, unfolding pathways. Like random switches discovered in Bob Blaker's lab, the conformation can be switched so as the function associated with each of the functions, conformations, by binding of metabolites. So one of which is a good mice ribozyme, which I will be talking about just a little bit. So this is a conventional view. So far there's no violation of this phenomenon. In other words, all these conformations, again, can be reversibly interconnected between all these conformations. So let me submit to you this hypothesis, at least it suggests the possibility of the following. What if we have one RNA sequence, and just for the sake of argument, that we have actually two non-convertible structures. They are not conformations because they are not connected by any thermodynamic pathways. So these are truly two different conformation structures. In other words, if we do a transcription from DNA to RNA, we can generate two different phenotypes. Each one of them can have its own conformation repertoires. So as usual. Now, as a result, what kind of consequences we can obtain by having this kind of a hypothesis for possibilities? First of all, transfer of different structures and associated functions by one sequence from DNA to RNA or RNA to DNA for that matter would be possible. So this would be different than from the case where another conformation would be the folding product of the ancestral sequence during evolution. Okay? And second, the two structures would share the same evolution history because one sequence will code for both, and the survival of one phenotype will ensure the survival of the other because of that sequence. And thirdly, the two phenotypes will always be present during evolution. And that will influence the evolution outcome. In the case you have two conformations, and one conformation or one function may simply be lost during the evolution simply because that particular conformation may be the folding product of the ancestral conformation. All right? So if I don't get through by all the times, then I will tell you, number one, this would be the summary slide that I'm going to give to you. And number two, I'm going to spend the rest of my time, hopefully, to try to at least generate or show you some of the data to demonstrate the possibility that one RNA can fold into two non-convertible structures. So these two RNA structures assumed by the same RNA sequence is actually an aptamer I'm going to present to you. And this aptamer or two structures act as a collaborating pair that inhibits the glutamate ion channel receptor. So let me pause for like a minute or two to try to explain to you what the glutamate receptor or the system I'm working on before I explain to you about the structure function of this particular RNA aptamer and the associated two structures. So glutamate ion channel is a ligand-mediated ion channel receptor. So glutamate is a neurotransmitter. In the absence of glutamate, the channel is closed. The binding of the glutamate channel opens, allows small cation anions such as sodium, small sodium or cations or potassium going through. In addition, a good portion of the glutamate ion channel receptors are highly calcium permeable. So the glutamate ion channel receptors are indispensable for brain function and development because glutamate channels mediate the neurotransmission and most of the excitatory synapses in the brain. And those receptors play critical roles in the synaptic plasticity. And the synaptic plasticity has been implicated as a cellular basis for certain forms of memory and learning. More importantly, if the receptors are hyperactive, they will induce the higher calcium into the cell, and the calcium overload in the neurons will end up causing the death of the cell. This will be a hypothesis or at least a mechanism underlying a slew of the neurological problems like ALS, epilepsy, and stroke, known as excitotoxicity. So one of the ongoing problems in my lab is really to understand the structure function of one particular subtype of glutamate receptor known as AMPA receptors. In addition, we're also looking at the structure activity relationship of various agonism modulator or positive negative modulators, and they are synthesized as drug candidates. And shown on this slide are some of the examples of various structures of these molecules. But the one thing I do want to mention is because they are small organic molecules and they have water solubilities, and many of them actually have cross activities either against different subunits in a particular family or subfamily, or actually have cross activities against subtypes of the glutamate receptors. So our goal, at least in relationship to the subject that I'm going to tell you today, really starts with an idea: is it possible we can generate a different type of template, such as RNA as an aptamers, as a new template, water-soluble, nanomolar affinity, perhaps have a better activity against a particular subunit or subtype of the receptors we're working on? The answer is yes. And the way we do this is by using the systematic elution of ligands by exponential enrichment or selects. And we can express the receptor in various forms in a recombinant way and also use the whole cell but in a membrane limited form and generate various sequences that are possibly the putative Aptamer candidate. In this case, indeed, after 14 rounds or so, I've found some sequences. And again, this library design with about 10 to the 15 variants and sequences came originally from Jack Schulstock's lab. Here are two pieces of constant regions on the 5' and 3' end. Here are the variable regions. Before I tell you what of the pieces I'm going to be talking about, let me tell you a little bit about functional assay. How do we look at the folding, and in this case, the effect of the folding or the function of those aptamers. And the way we do this is really by electrophysiology. Because glutamate receptors are ion channels, we can express it HEK293 cells and do whole cell recording, and we can apply solution in the presence or absence of aptamer and measure quantitatively the amplitude in the presence or absence of this, and as a way to measure the function of those RNA pieces. One particular RNA or RNA aptamer we've identified is the AM1444. We've done the truncation from both the 5' and 3' in order to identify a minimal, a functional sequence. Finally, we did. This would be a 58 nucleotide sequence. And what is interesting now, we cannot touch the 5' end. We're going to lose all the function. But we can sequentially do the reduction until we find the 58. Anything shorter than this, we will see a loss of a function altogether. The bottom line is the IC50 value for this particular piece, or AM58, strikes to or reach to about 30 nanomole. A particular subunit of AMPA receptor, which is ROD2, and this particular aptamer is perhaps the most potent inhibitor ever documented in literature. So, that's not a story as much as it is regarding the aptamer. Now I'm going to turn into the attention what happens to this particular molecule or aptamer. So if we run a native page, if we do all kinds of the sequence reduction, we also find multiple bands, including at the very end of this minimal functional sequence at AM58, we saw two bands. We will call it upper one M1, we'll call it lower one as M2. What is interesting is if we never separate the two pieces, it is a very potent inhibitor, like I showed you before. But if we carefully isolate this and purify this and test it on the cell containing or expressing the receptor, we saw no inhibition for each one alone. But when we mix it with 1-to-1 ratio, we can fully rescue the inhibitory function. So they have to work as a pair. For the time that I won't have to explain to you for a whole lot of this, we actually established the ratio of M1 and M2 the work is 1-to-1. This would be a competitive inhibitor, etc. What is also interesting is the following. Here's a series experiment demonstrated that M1 and M2 generated by the same transcription reaction will actually bind to two different places on the same receptor. For instance, if we use a RM87, which is totally non-functional by its own, like shown here, it would not have any inhibition if we mix with M1 like this. So the ratio 1 indicating no inhibition whatsoever. And if we choose 53, so we make sequential reduction, we remove 5 more nucleotides from the 3' end, and we can see that AM53 would be able to inhibit the receptor just as good or as well as the M1 and M2. Yet the AM53 is not able to combine with M2 to form any inhibitory effect whatsoever. So this would indicate AM53, by the way, alone doesn't work because it's not functional. So what this suggests is M1 and M2 would bind to two different places, and the 53 still inherited some sort of a structure just like M2. Now, we have additional evidence by binding, by other electrophysiological evidence to show that, again, M1 and M2 binds to the receptor by 1-to-1 ratio. So that's kind of the idea we want to do. So, again, because of the time constraint, I want to go through to you. we know that M1, M2 acts as a competitive inhibitor. So the way we believe it works is that they have to bind close enough to cause a stereo hindrance to prevent glutamate from binding to its agonist site as a competitive inhibitor. So that's as much as it is about the inhibitor. So what will actually happen? We saw the two bands. This will be one sequence. So do they represent two different structures, or is one of them is a folding product of the other? So that's the beginning of this intriguing question. Now, M1 and M2 are not interconvertible. That's or at least the hypothesis I've brought to you at the very beginning. And this is a piece of an experiment done by most using the denature and refolding experiment. Because we cannot denature it, so I'll tell you this refolding literally is not a problem. Well, we have done the following. M1 and M2, if we don't purify this will show where they are. If we carefully purify M1 and M2, and this shows where they are. What is also important, this is a native gel, and if we run the denature experiment by using the saturating urea, by boiling for 15 minutes or so, and also in the presence of more than 50% of formaldehyde, as in many of these denaturing conditions you can possibly think of, what we have found is the following. M2 stay where it is. M1 partially degradated into something, well, not folded into something. But the mobility on the denature gel seems to be identical. So we figured the reason that if this piece were lower than part of the M2, then upon renaturization, and this piece would stay where it is. And in fact, it was gone. So therefore, we conclude this tool has absolutely, after many years, or at least 4 years of work now, we have failed to identify or find any denaturing condition whatsoever to be able to interconvert between M1 and M2, vice versa. What is also interesting is that if we use a glycol to modify or destroy all secondary structures, we see they are identical. And again, if we use this good old method of using NaOH, you shot the sample at the room temperature for a very brief period of time, like 5 minutes or so. Either it nothing happened, or we can totally degrade the sample, but yet we have failed once again to be able to interconvert both species at the same time. So on that operating assumption, at least unless we suggest there's additional things we can try, so I'm going to claim that M1 and M2 at this moment are really not interconvertible. So this is not a RNA folding product. M1 or M2 is not a folding product of the other, but rather they represent two different phenotypes. So, and I would suggest that M1 and M2 will be made during transcription. So this is a co-transcriptional product. And this gel shows a typical standards of this enzymatic reaction. In other words, the yield of M2 and M1, M2, and the rate of M1, M2. Obviously, M2 is made faster. At all times, it would be influenced or affected by pH, by temperature, by magnesium concentration, etc., etc. Indicating or suggesting these results are consistent with the fact that M1 and M2 are indeed the transcriptional product. To make the story a little bit juicier, so we've been able to make this synthetic AN58 from commercial sources. So these are totally made by the chemical synthesis method as a control field. What is interesting, if we run them in the same place, native gel, you can see that the synthetic made AN58, number one, is a homogeneous population. The mobility is different from either M1, M2, actually it's low. Functionally, synthetic AN58 is about 5-fold more potent than the combination of M1 and M2. So for the rest of the talk, I'm going to use this as a control field. So first of all, we've shown that M1 and M2 has the same sequence as reviewed by using the dideoxy-mediated RNA sequencing. Now you may argue that the last 15 nucleotides from the 3' end is actually low. Okay. And then what happens is that we're going to try to see, so you may argue that the 3' end is not clean. So we've used this PRAV23 construct, which we obtained from Bob Beatty's lab from UC Colorado, and we made this AN59 aptamer by using this green mass web design containing construct. So this work is originally reported in Breaker's lab. So what happens when we insert our sequence upstream at the AG site, including the A, just in case to drop this. So we can use this again just to show this. Now we've made it. Just in the sense by using LC mass, you can see that AN59, including 1A, is perfectly identical with M2. We're using synthetic control to be exactly the same molecular weight difference by that 1A. And AN59 M1 M2 is exactly identical not only in its denaturing protocol, but also in the functional test assay. Very quickly. And we ran some shape experiment, you can see they are actually different in a sense, 3 of them. By building all of this, and we can see that at least by using reactivity-wise, are different. So we've done some, I'm going to fast forward to keep the time constant, that we've done additional enzymatic and chemical probing and shown that the 3 of these structures are absolutely distinct. What is also fascinating about this piece, because it would be most or best fit by this conventional nearest neighbor free energy minimization model, and the free energy is actually about 16 kilocalories per mole different. That, in other words, be more stable than the result or the constraint we obtain from chemical and enzymatic modeling. And right now we're doing some 3D modeling collaboration with Ross Altman's lab at the Stanford University. So I'm going to kick out of this, probably stop one slide in this case. That now, if M1 is not a folding product of M2 and vice versa, so where do they come from? How do we select this from billions and billions of structures? That would be too coincidental. So here's the thing that I want to allude to you. In other words, how have M1 M2 been evolved from this vastly different sequence population? The idea is the following. The probability of a species to be evolved will be proportional to its concentration during the evolution. But in this case, the survival of any one of the 2 species, in this case, will ensure the evolution of the other. So the other species never goes away. So if M1 and M2 are not the folding products, then they must be that no chemical denaturing can defold them. Then the reverse transcriptase will have to be the denature, because we run the selects. This is literally the in vitro transcription experiment. So this is the last slide I want to show. And again, we have done 2 experiments. First one, that you can see M1 and M2 using synthetic control. Both of them can be fully reversibly transcribed. What is also interesting, if we take the RNA M1 M2, we purify this, we run RT-PCR going backward into cDNA, and then we show the same value. If we run the transcription again, we can fully generate both M1 and M2 from pure subpopulation of either M1 or M2. So that suggests both of them actually evolved from this condition. So finally, I won't have time to go through all of this, but again, the idea is really using this epochal evolution cycle and showing that according to this cycle that suggested back in the late 70s, that Kamara hypothesized this evolutionary view about evolution in the sense that each of the sequences can diffuse into a neutral network by mutation, but it is implied that only one sequence is correlated with one phenotype. But in our case, because one genotype codes for 2 phenotypes, so it was almost impossible to visualize, except you would be able to see that between 2 networks, neutral networks, the evolution must go between, at least for some period of time, in between these 2 networks in a sense. So I'm going to stop here. The last slide I really like to show the following. I like to thank Dr. Phil Sharp. I don't know if Phil remember this. And about a year and a half ago, I approached Phil, and Phil has given me really valuable advice on how to approach this problem. And I also benefited my interaction with Ron Breaker, who gave me the advice on running E9 protein, which I didn't show, but we published. And Ron suggested using the glutamate ribozyme conconidase, and David Schum and Eric, who's not going to be in this meeting, have given us the actual cellular portion of the soluble portion of the glutamate receptor for some of the studies that I showed before. Also, let me thank David, Sunyali, and Tom for all the discussion and suggestions. And finally, I thank you for your attention.